Let's see. Okay. Collaborate. You're just gonna work with me today. Okay. Can everybody see this roadmap slide about vision? Is that showing up okay on our Collaborate?
Yes, we can see it.
Yes. Thank you. Wonderful. All right. Let's get rocking and rolling then. So on Wednesday, we left off and we've been having sort of this conversation about what traits allow our fishes to be successful, or any organism really, right, to be successful underneath the water. And so we've been talking about several different features. So first we talked about respiration and the challenges that water presents in order to get oxygen out of the water. So we talked about things like viscosity and low oxygen content, as well as some other issues like salt and temperature, and how particularly fishes use things like some very high surface area gill structure, as well as that high efficiency countercurrent exchange, where we can have exchange between the water and those capillaries within that secondary lamellae at every single stage they're in contact with one another to be very efficient. We also talked about several different ways that fishes can adjust their buoyancy, from using livers, heterocircle tails, right? We talked about 2 different kinds of swim bladders, depending on that habitat of the fish, which really then depends on how much access to air do I have in order to fill that swim bladder. Right? That swim bladder is kind of like a balloon inside their stomachs. And then at the very end of class, we started talking about actually getting sensory inputs, right, which, due to the nature and ultimately kind of that viscosity again of water, presents its own challenges. So first we talked about mechanoreception, which is a bit like touch or hearing, if we kind of pool them together. So we looked at things like the lateral line, which looked at vibrations. We pulled this in with the inner ear, or what fundamentally is the inner ear, right? So we looked at things like the semicircular canals, as well as those otoliths, those little ear stones. We also looked at electroreception, right? And this is largely, but not exclusively, on our chondrichthyes. And we looked at an osteichthyes example of this as well. Right, so remember, largely we talked about the ampullary receptors. So an example of that would be, right, the ampullae of Lorenzini on our sharks, the thing that kind of looked almost like acne all along its entire head. These were pits with nerves that allowed them to detect electrical signals, right, but very small ones, right, like heartbeats and muscle contractions. So before we revisit vision, were there any questions about any of those other things that we had talked about for our fishes? Okay, so at the very end of class, ooh, rude, I know I picked 10. There we go. On Wednesday, we had started talking about vision. So finally added some sight to the conversation. And in this case, the first thing we talked about is how light refraction, right, which we sort of defined as focusing light, differs significantly for underwater organisms than it does for land organisms, right? And this is a little intuitive to us, right? If you dunk your head underwater, you can see, at least, right, superficially, right, if you don't go too deep, but your vision kind of sucks. And so we want to think about, well, why does that happen? And so the first thing we talked about is, well, if we look at the human eye, we see that we have this big round cornea, right? And we know this if we think about our own eyes, right? That this front space, right? That front shiny, clear area in our eye is quite rounded. And that's because that area, that cornea, remember, is doing most of the light refraction for any terrestrial organism's eyes. Okay, meaning that the front stage is doing most of the focusing when light enters your eyes. And remember, we talked about this is why humans can get LASIK, which is basically a laser coming in and reshaping or resurfacing that cornea so it does a better job focusing light, so then you don't have to wear glasses or something. Once light passes through there, so that does upwards of 85% of your light focusing, colors here, then okay, the lens, which is the second rounded item, sort of does the rest, right? This is supplementary focus. So by the time the light then reaches your nerve, right, this is what carries the image to your brain, everything should be perfectly happy, snappy focused. Okay. The reason your vision sucks underwater is because water also bends or focuses or refracts light. It's a dangerous word to be saying wrong. And water bends or focuses light in the exact same way that the cornea does. And since when we go underwater, we don't really have anything else that does a lot of work. Remember that lens only is quite small and it's only doing maybe 15% of the work. There isn't a whole lot of additional focusing that our eyes do once we're underwater. This is why if we look at our fish, switch back to red. Okay, if we look at their cornea, their cornea is quite flat, right? It's compressed against the rest of their eye. It's almost non-existent. And this makes sense, right? If we just said, well, the cornea is doing the same kind of focusing or light refraction as the water, well, then there's no point really in investing or making a large cornea because it's not really going to do anything. Okay, so what we see instead, let's switch back to green, with our fishes is a much larger lens. Okay, here we can see that it's quite large and substantial, very round, right? This is where we left off on class on Wednesday, where we can see that secondary stage for us is going to be the primary stage for any organism that's underwater, right? Because it can do the bulk of the light refraction, right, the light focusing, because it's doing a different kind, right, a different step of light bending. Okay, so in this case, right, this is doing most of the work. Okay, and so you can tell that because this is where the largest feature of the eye is. So if we can just glance at the eyes, by looking at which is the largest piece, you can tell what's taking on the bulk of that focusing. Okay, I think that's where we left off on Wednesday, but I wanted to make sure we kind of had a chance to go back and review since we left in sort of an awkward location. So there are several other pieces that are certainly going to affect any organism's ability underwater to see. So even if we had fish's eyes when we went underwater, there's still a lot of struggles that we would encounter, right? And a lot of this has to do with light itself, right? Vision is highly dependent on the light that we encounter, even if we're not seeing color, right? Because color and light are associated. So if we think about going into the water, right, the deeper in the water that you go, right, the less light in general that's going to penetrate, right? So the deeper you are, generally the darker that it's going to be. So that alone, that's what we're looking at here, that alone is going to have a negative impact on how well you see. And this is something we saw a little bit with our early fishes, right? Our hagfish and our lamprey, who spend both, right, a large proportion of their time in very, very deep water. Remember from lab, we saw some videos, for example, of our hagfish feeding on that sunken whale carcass. So while we could see that video pretty well, that's because the submarine that was taking the video was putting either infrared or dark light cameras on them. Normally that space is extraordinarily dark, right? That's the bottom of the ocean. So when we looked at those organisms, right, a lot of those organisms didn't have true eyes. So when we first handled our sharks and our fishes, seeing like real big eyes was almost like shocking to us compared to what we've been seeing, right? In fact, our hagfish largely only had things like the pineal gland, right, and these light dark sensors on their body, because for the most part that's all they were seeing. And you see a lot of that in these deep water fishes, because why invest in these big beautiful eyes, quite frankly, if they're never going to be used for anything. The other thing that we're going to see as we go deeper is it's not just overall light, right? It doesn't just get darker as we go deeper, okay? But things like color, right? So if we look at the image we have on the right here, does not equally get lost as we go deep, right? Different wavelengths of color are able to penetrate deeper into the water. So for example, as we look at our wavelength split here, let's grab some different colors here. I see right away things like my really warm colors, right? So my reds and yellows. These colors don't penetrate very deep. Okay, so these are not colors that go very deep. They cut off very quickly, usually somewhere between 30 and 50 feet, and those are not colors that are going to go any further, okay? The colors that do tend to go very, very, very deep, right, are my very cool colors. Okay, that does show up better on your screen than it does mine. Right, so we see a lot of our greens, and in particular our blues, right, are going to come very deep into the water column. So we're going to have darker light, okay, but a specific type of light. And this is part of the reason that you don't see as much algae getting all the way down, right? And there's also a lot less DOC. So if this is a new term, DOC just means dissolved organic carbon. Okay, so what this is basically telling us is because you're not getting as much light penetration and you're not getting a broad range of colors for light penetration, that pretty much anything that's getting past that 30 to 50 meter mark, you're not going to get a lot of algae and phytoplankton that can live down there. Photosynthesis is much harder. And most organic materials, right, things that organisms feed on, are not going to be drifting down there. Okay? And this is something else we kind of saw in that hagfish video, right? Food towards the bottom of the ocean can be quite scarce. So when it does show up, like that whale carcass, it's kind of like a feast situation, right? Everybody shows up all at once, they all snag it while it's there, and then it kind of goes back to being this desert again. Okay, any questions about the challenges that you would experience for vision? I just feel like I'm doing a little bit of like a Blue's Clues episode when I'm doing some online classes. Okay, let's see if I can, how do I clear this? Can I just go? Will it just leave it? Yes, okay, I like this feature. Okay, so while vision is really helpful, we know a lot of fishes and stuff do have eyes, and a lot of these eyes are going to be very specific, right? We're going to have large lenses, or maybe I'm just going to be able to focus on just blues or just reds, depending on where I'm at. As we noticed with things like the lateral line and the otolith, or with electroreception, just relying on vision is probably a faulty situation, right? Because it's not going to be wholly dependable depending on where we live. And so we want to keep adding different types of sensory features. So I have a fallback no matter what kind of habitat I'm living in or what happens to my habitat, right? And even as humans, we know vision is not always something that we can trust, right? So another sensory system that we can rely on is called chemoreception, right? Which pretty much means smell. Okay. Their sense of smell and our sense of smell, oops, that was quite loud, I apologize, is very, very similar in its functionality. Okay, so inside your nose, right, if you've ever looked inside it or felt inside it or seen images, and we'll talk about this more later in the semester, a lot of this has to do with having a space that has lots of little bony protrusions, right? And so remember, one of the big themes here is the more surface area that you have, listen, I'm clicking, you got to do the thing. Right, the more surface area you have, the better you're going to be, right? So we have a little bit of surface area in our nose, our smell is right? Something like a bear or a wolf has a lot of surface area, right? Lots of little bony protrusions in their nose, so they're going to have a really good sense of smell. This is the same thing we kind of see in our fishes, okay? So if we look here, and I've got it blown up here, of course, they also have little protrusions in their nostril space, okay? Their protrusions aren't bony, but instead soft. Okay, they're made out of soft tissue, which is what this rosettes term means. Okay, so they're made out of basically skin, right, epithelium. But fundamentally, the structure is exactly the same. So we see here, okay, soft tissue protrusions. Okay, and all of these are going to function the exact same way that our bony protrusions do. Okay, the more protrusions that you have, right, so the more folds, right, the more surface area that you have for nerve endings, right, these sensory receptors that can accept, right, these smell particles or sense particles that are floating in the water. Okay, the more particles, right, that can land on receptors, the better sense of smell that you're going to have, right? And so there's certainly going to be variety within fishes. Okay, so a fish, for example, that spends most of its time in very clear open water, right, something like a tuna fish or a boxfish, right, they spend most of their time patrolling open water, they're probably going to have less. Okay, but some kind of, man, my throat sucks today. Some kind of fish that's going to be spending its life in a really murky or turbid pond, for example, so something maybe like a catfish, right, they are always kind of digging around at the bottom. Their vision is going to constantly be clouded either from like floating mud or from their pond itself being very dark and murky. Okay, they're going to have a very good sense of smell, right, because they're going to use this largely to navigate their space and to hunt prey. Okay, in the same way that we see the same variation in our terrestrial organisms. Okay, any questions on smell? Okay, so just like when we saw this for respiration, one of the big keys here, right, is we want to get continuous fresh water flow, right? So it's kind of like snorting water through their face. So we can constantly get new smells. It's like tracking, right, with their nose. Right? Very relatedly, just like when we're smelling something, right? Oh, that smells so bad, I can taste that. Or, oh, it smells so good, I can almost taste that. Smell and taste is also tightly related in fishes, right? Your nose and your mouth are connected, right? This is why you need to wear the mask over your mouth and your nose, right? They're connected respiration-wise, and they're also connected palate-wise, right? The way you taste things is correlated. And so taste is also connected with chemoreception. Okay, taste works the same way for fishes as it does for humans, right? So we use this both for food recognition, right? I taste something, it tastes good, it makes me want to eat it. I know I should and can eat it, right? So for example, sharks, right? Most shark attacks that you hear about, right, are not a shark ate a whole human, right? They're usually sharks bit a human, and maybe the human dies, right? Or the human gets away. Okay? But usually if the human dies, okay, it's because they've bled out, right? Because a shark bite's kind of a big deal. Sharks are quite large and their teeth are all sharp and pointy. But usually what's happening here is called a test bite, okay? My husband calls them no thank you bites, right? If you're a human. So the idea is you're trying something. So this is kind of what we see up here, and this is what's happening with sharks, right? Is you see something that looks a bit like food that you recognize, right? And so I'm going to take a bite on it, right? Usually if we're talking about humans, that's going to be on a leg or an arm, unfortunately, right? Sort of important appendages. So in surfer attacks, sharks are taking test bites, okay? We don't taste terribly good, okay, right? Particularly when you think about, for sharks, their core food source are things like sea lions, right? This is a major food source, okay? We are not blubbery, right? Or we don't have a good oil content or fat content to our meat. So we're a very disappointing food source, and usually are not going to continue to be pursued. That still is not necessarily great news. Now we're in the middle of the ocean and bleeding out horrifically, right? But still not a core meal source. Okay. The other thing that we see for food, of course, is deterrence for toxins, right? So I taste something or bite into something, it tastes very bitter, it tastes very bad. Okay, this I know then means I should not be eating it, right? You taste very bitter leaves or bitter berries, makes you question whether you should be eating it, right? I taste something bitter from my fridge, tells me maybe it's gone bad. Let's not eat that, let's throw that away. Okay. So taste is a little more intuitive, right? This is functioning very similar to the way terrestrial organisms, right? And in particular, in this case, the way humans use it. Okay. That finishes up all of our sensory organs and sensory challenges. So are there any questions on any of that before we move on to talking about osmoregulation? Okay. Just want to do kind of a quick vibe check. Everybody's still there and can still hear me, right? I'm kind of feeling just this side of crazy right now.
Yeah, we can see.
Thank you so much. Okay. Okay, so let's finish up our life in the water conversation by talking about osmoregulation. And so when we're talking about osmoregulation, we really kind of want to picture organisms like a leaky bag of water. Okay, so now remember, when we're talking about osmoregulation, we're talking about saltwater balance. Okay, and so we'll look at different kinds of salt when we're talking about that. Okay, that's ultimately what it comes down to, right? I need to make sure that my body has a good amount of salt and water to keep me going, right? I can't be 100% water and I can't be 100% salt or I'm going to die. So how is it that as I move around, okay, that I can maintain that, and ultimately, usually, although not exclusively, as we'll see, okay, I need to have a different salt and water concentration than the space that I'm living in, okay, which is key and really important. All right, so let's start by thinking and having to review a little bit of diffusion principles. So I bet we didn't think that those were ever going to come up to haunt us again. Okay, so the first thing I want us to think about, so let's take a hot second. Here we are. Here's our organism. Okay, so I have a very specific salt concentration, right? So these units here that I have, let me draw, oh, causing drama. No, of course not. Click it twice. Ooh, blue is a terrible color. Let's not choose that, huh? Try yellow. There we go. These units are just saying that is the concentration, right? So what I'm saying here is my body has 300 units of salt, okay, per liter, and I'm living in a solution, right, this blue space, like let's say the ocean, okay, and the ocean has 1,000 units of salt per liter, okay? So if I take an organism that's at 300 and just plop, drop it into the solution or the ocean that's at 1,000, okay, what is likely going to happen? So go ahead, take like a hot second and think about it in your notes. What is going to happen here? Or what would happen here? Think about all your diffusion properties. I know it's been a while since you maybe have had to work with that. So let's think about our rules of equilibrium many, many moons ago, right? So if I think about my rules of equilibrium, what generally wants to happen here?
The concentrations of salt water would want to be equal.
Right, exactly. So I want the concentration of salt water, the concentration of my person is going to want to be equal here, right? So that's going to mean two things are going to happen, right? The water from my person can go out via osmosis, right? And the salt concentration here can go in. Okay, and so we're going to look at in a second here what exactly can happen, and of course we would want to prevent that from happening, because that sounds sort of bad from an organism's perspective, right? If I drop you in the ocean, you would not want all the water to leave your body and a bunch of salt to enter your body. But that's fundamentally, right, what we know wants to happen, right? That follows our laws of diffusion and equilibrium. And so from an organism perspective, that's like super bad. So as we think about osmoregulation, what osmoregulation is all about is how does that like not happen? So we have two choices as an organism to get around these issues. We do have the choice, as we see, is this top bullet, colors because I changed it to yellow now. That you can keep your internal conversation gracious, concentrations the same, right? So you can just allow it to happen and let all the water leave, and we're just going to let equilibrium be a thing. And that's called being an osmoconformer, right? So we can break this word up, right? So conform would be, I'm going to be the same, right? And osmo refers to that saltwater business. Okay, we don't see this a ton in vertebrates, okay, but it does happen. Right, you have some jellyfish that do it, some crabs that do it. Okay, there are organisms, in particular organisms in the ocean, that live their best life and do these things, right? So it can happen. And you can imagine how it's sort of like the easy choice, right? Let the ocean take over, right? I don't want to be in control of my own life. And I can sort of relate to that some days. Okay, but more commonly, as you might imagine, there's some problems with that concept, right? As the salt water balance around you changes, right? It is maybe a dangerous choice just to let the environment control your life. So more commonly, and what we spend most of our time talking about is regulating or being in control. Okay, I want my body to be different than the space that I'm in, okay, for any number of reasons that we'll talk about. So we call this situation an osmoregulator. Okay, so here again, we see that that prefix for salt water. Right, and I'm going to regulate it, right, or I'm going to control. Okay, so let's go ahead and take a couple of minutes here and really explore, what do these two different options look like? Okay, and what are some of the inherent problems or struggles with each of them, right? So why wouldn't everybody just be an osmoconformer? Because it really does feel like the simpler solution. And we know, right, that an organism has a finite amount of energy, right? I don't have an infinite amount of energy to do everything. So if I didn't have to invest energy, for example, in doing osmoregulation, I could put that energy elsewhere. So why don't I just do that? Okay, any questions so far? These definitions feel okay? So let's start by looking at osmoconformers. Okay. So if we think about the example that we started with, I've gotten very lazy with what my organisms are. Inside my bubble, right, is an imaginary hypothetical organism. So let's say that's a jellyfish. Okay, and this is an osmoconformer, right? So this is an organism that's just going to let the environment take over its life. So here we are in the ocean again. Okay, I need to be yellow again. Right, my ocean has that 1,000 salt units per liter. Okay, very salty. Right, and so the jellyfish is just saying, you know what, fine, this is fine. Okay, I'm also going to let myself be. Okay, I'm basically going to be a floating, leaky Ziploc bag. Okay, and that works out fine for the jellyfish. Okay, but let's explore what some of the potential problems for this are, right? So let's look at a different osmoconformer situation. What if, for example, I wasn't in the ocean? Okay. So in this case now, I'm in a freshwater situation. So most freshwater, and this is not exaggerative, right? Most freshwater has near zero salt, right, of actual salt, right, sodium chloride. So if I am an osmoconformer, right, so for example, what if I chose a hydra, which is sort of like, I guess, a miniature version of what a jellyfish would be. If I'm going to be a hydra or an osmoconformer in this situation, right, I would also have to have 0 units of salt per liter in my body. Can I do this?
No.
Doesn't all bodies require like a certain level of salt?
Excellent, right? So I can't do this, right? So this was okay in the ocean, right? Because you can just sort of adjust to have having a higher salt content. But your body needs salt. It needs some kind of salt, right? Sodium, chloride, calcium, these are all salt ions, and they're used to make your muscles move. They're used to make your heart pump. They're used in basic cellular processes. So everything that happens in a body has to use some kind of salt. So we can't have no salt. So we find in a lot of conditions, and for certain bodies as we'll find, that having osmoconformity is just not possible, let alone realistic, right? Because you wouldn't actually have a functioning body. So it's under these parameters that we find having some kind of system in place that allows you to be an osmoregulator becomes very important. Okay, so let's look at how can we make ourselves osmoregulators. Okay, so any questions about anything we've talked about up to this stage? Shiny. All right. So as we look at how we can regulate ourselves, there's several different techniques that organisms can use. In this case, I'm specifically talking about fishes that can help regulate saltwater balance or help prevent loss and exchange of water and salt ions. So we're going to talk very generally about these 4, and then at the end we'll have some examples about how they're working. So first and foremost, we see that we have scales on here, right? And this can be any type of scale, right? So if we talk about our sharks, remember, right, we have to kind of remind ourselves that even though those scales were flaking and flicking off and sticking to the sink when we were handling them, that they definitely did have a very specific type of scale that, you know, dulled our scalpels when we were cutting through them. So any type of scale on these fishes, in addition to being, you know, a type of protection from abrasion or battle, also works for impermeability, right? This is the term we see on here. Okay, meaning that this is a layer over the top of their skin, right? So like if you think about our skin, where's the camera here? Our skin is really terrible for exchange, right? But our skin has a different job. So your skin, for example, gets very dried out very easily, right? Not judgment, but very generally. So we have a very easy loss overall for moisture, okay? But our skin is also there in part for heat and gas exchange. They don't want that to be happening. So they have another layer over the top of their skin. So when their skin comes in contact with that, you know, 1,000 or 0 units of salt, right, that diffusion is not just going to easily happen. It's not going to run dead stop towards equilibrium. So instead, they've created a wall, right, or a barrier in between their saltwater content and the saltwater content of the space around them. So diffusion is not going to just immediately take place. Ooh. Okay, pardon my handwriting. Okay, so that's a great first step, right? The moment we can stop that exchange from barreling towards equilibrium is one really good step for us. Okay. The next thing we see, okay, are what are called chloride cells. Okay, so remember when we're talking about salt, I'll pull out some chemistry, right? Salt is sodium chloride. Okay, so when we're looking at chloride cells, okay, these are a very specific type of cell that are present in the gills of the fish, right? So this is part of their respiratory organ. So remember, we breathe in, and then we swallow and push out through the gills. Okay, so in the gills then, we have very specific cells that are focused on actively transporting chloride. Okay, now which way it goes depends on the fish's environment, right? So if I'm a freshwater fish, okay, and there's not a lot of salt in the environment, okay, I'm going to work at keeping, right, because it's already going to be very hard to find. So I'm not going to want to like kick that back out into the environment. We're going to work on maintaining, right, or keeping all of that extra chloride. It's going to be very difficult to just find that swimming around, right? If, for example, I'm a saltwater fish, okay, there is salt everywhere, right? Too much salt. They're drinking salt and breathing salt, and literally salt is getting in their nose, and salt is everywhere, right? I can smell and taste it. They do not need salt extra in their diet, right? So if I'm a saltwater fish, right, we are going to ditch it. Right, so I can actually then excrete some salt through my chloride cells. Okay, now you don't have fish jumping around from one to the other, so my chloride cells is going to do one or the other. So if I'm a freshwater fish, right, these cells are set up to keep my chloride. If I'm a saltwater fish, these cells are set up to get rid of. On Monday, we will finish up all of our life in the water by talking about pee, which will be the very last topic here. You see it's the last bullet sets on here. We're going to drink and pee. I'm trying to get the color exchange. There we go. I don't know if I actually pushed start. I did not. There we go. So your socrative is zeroing out. There we go. Your socrative is now up. So we'll finish up by talking about P, which has a lot to do with the nitrogen, which is another type of solute that we need to make sure that we can exchange. And then we'll go through some examples on how we can be osmoregulators, and that'll finish up all of our life in the water. kind of shrinky dink this hopefully and see the chat. Okay. While we're finishing up our Socrative, is there any questions before we finish up class?